Good afternoon, everyone. I think it's time to get started. So thank you so much for joining us for the 19th annual York Gairdner Lecture. I want to start by acknowledging the land we are on today. York University acknowledges its presence on the traditional territory of many indigenous nations. The area known as Toronto has been taken care by the Anishinaabeg Nation, the Haudenosaunee Confederacy, and the Huron-Wendat, as well as the Métis. It is now home to many indigenous peoples. We acknowledge the current treaty holders, the Mississaugas of the Credit First Nation. This territory is subject of the Dish with One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. York University is honored to serve as host yet again to this year's Gairdner Lecture. As a leading research and teaching institution, York University is a force for positive change. Ranked in the top 40 universities worldwide in the 2023 Times Higher Education Impact Rankings for advancing the United Nations Sustainable Development Goals, our community of change makers, from faculty to staff to students, are all united by a commitment towards building a better future. Today, as hosts to the Gairdner Lecture, we welcome, learn from, and celebrate Canada Gairdner Awards laureates who are similarly united by that same commitment. Each Gairdner Lecture held across Canada provides us with the opportunities to hear from the laureates whose work has a profound impact on the health and healthcare outcomes of global populations through the discovery, application, and translation of biomedical and global health research. This lecture series grants us access to the scientific knowledge and the chance to hear from truly brilliant minds. Since 1957, the Gairdner Foundation has inspired scientific excellence all over the world with mission to recognize and reward groundbreaking research that impacts human health. To date, 418 awards have been presented to laureates from over 40 countries. And recognized as one of the most prestigious prizes in health research, many of the laureates go on to receive the Nobel Prize. In fact, several Nobel laureates have been York Gairdner Lecture speakers. In fact, one of the last year's Gairdner Lecture speakers, Dr. Caitlin Carrico, the 2022 Gairdner International Award winner, was named the Nobel Prize winner in Physiology or Medicine, along with her colleague, Dr. Drew Weissman, for their discoveries that enable the development of the mRNA vaccines against COVID-19. What a pleasure it was to host Dr. Carrico at York last year. Today, I'm honored to welcome two outstanding scientists to York University, Dr. John Jumper and Dr. Deborah J. Cook, whose distinguished work sets them apart in their respective fields. Dr. Deborah J. Cook is the recipient of the 2022 Canada Gairdner Award for pioneering research that has developed and defined evidence-based critical care medicine in Canada, informing best practices around the world. Her 30-year contribution has greatly alleviated the human and economic costs of critical illness for patients, families, healthcare systems, and society. Let's give her a round of applause. Dr. John Jumper has received the 2023 Canada International Gairdner Award for developing AlphaFold, an artificial intelligence or AI-based solution that can predict the structure of a protein, providing the most accurate and complete picture of the structure of all proteins in the human body. This has immense potential to accelerate biological and medical research, and the solution has already been accessed by 1 million researchers and users in more than 190 countries. we are honored to have both Dr. Cook and Dr. Jumper to join us today to share their groundbreaking work. Dr. Cook, Dr. Jumper, thank you for your contributions to research excellence and for joining us today at the York Gairdner Lecture. Before I pass on the speaking arrangements to Dr. Ron Pearlman, I would like to thank a few people. An event of this nature is not possible without the help of volunteers who did all the work. So I'd like to, if you could just rise as a speaker names, Bob Edwards. Bob. Jen Steves. Tina Saputakis, is she here? She's outside. And Ron Pearlman, of course. This has been Ron's idea right from its inception 19 years back. And I can tell you that Ron has always been there in all of the 19 presentations we had, Ron. Is that true? So, and we hope that you can continue with working with us on this great initiative in the years to come. So I would now like to welcome Dr. Ron Pearlman, who is going to do further introductions. Thank you.
Okay, thank you and welcome. I'm not going to take any more of your time, or very little more of your time, just to thank everybody for coming and for being able to do this again and have these wonderful speakers to present to us. So what just a couple of brief comments. One is this will be recorded, and so we will let you know when and where it will be available. We don't know that yet. And just to ask the speakers to, because of the camera that's being used, sort of stay within 2 seats of the podium, just so we make sure we have the images of both slides and that. So now I'd like to ask Brittany Remnant, who's a first-year master's student in biology, to introduce our first speaker. And I must say that she is one of the people at York, and I'm sure there are many now, who are using AlphaFold, but more than using it, she's helped me, and she realizes it's an unenviable task because it's hard to teach an old dog new tricks, but she has been teaching me how to use AlphaFold, and I very much appreciate that. So, Brittany, thank you.
Hi, everyone. I'd first like to echo Dr. Asif and Dr. Pearlman in welcoming you all to the 2023 Canada Gairdner Lecture Series here at York University. My name is Brittany Remnant. I'm a biology master's student here in Dr. Mark Bayfield's lab, and it's my honor and privilege to be introducing our speakers today, two remarkable scientists and Canada Gairdner Award laureates this year, Dr. John Jumper and Dr. Deborah Cook. Now, before introducing our speakers, I would like to acknowledge both the Gairdner Foundation and York University for gathering us all here today to celebrate scientific inquiry and discovery. I was fortunate enough to complete my undergraduate studies here at York, where the university and faculty encourage students to engage in scientific research. I held Dean's and NSERC undergraduate research awards and also completed my honors thesis project here. And it was experiences like these, along with the opportunity to attend academic conferences and lecture series like those hosted by the Gairdner Science Week, that really solidified my decision to want to continue my academic journey in science. So I would like to thank our speakers in advance for being here today, because it's lectures like these that continue to inspire scientists at all levels of training to continue to be curious and work hard to make a difference. So with that, it's my honor to introduce our first speaker. Dr. John Jumper is an esteemed scientist whose pioneering work in artificial intelligence has revolutionized protein biology. and he's a recipient of a 2023 Canada Gairdner International Award. Dr. Jumper received a master's degree in physics from the University of Cambridge and earned his PhD in chemistry from the University of Chicago, where he developed machine learning to model protein dynamics. In 2018, Dr. Jumper became the DeepMind AlphaFold research lead, where he and his team aim to utilize artificial intelligence to predict the structure of unsolved proteins with atomic level accuracy. In 2020, Dr. Jumper's team achieved this goal with the development of AlphaFold 2 and were recognized in the 2020 CASP structure prediction competition as the solution to the longstanding problem of protein structure prediction. And the implications of being able to accurately predict protein structure can't be understated. With the limitations set by an experimental structural determination, only a small fraction of proteins have actually been solved. And given that protein function is directly tied to structure, developing a way of accurately predicting how proteins fold provides insight on a greater number of proteins to a greater number of people. And additionally, the choice to make AlphaFold open source has and continues to support the understanding of disease, therapeutic targeting, drug development, environmental conservation, and support of basic science research. And in this room alone, I suspect that many individuals who work on proteins with unsolved structures have turned to AlphaFold for some more insight. I certainly had the pleasure of using AlphaFold during my undergraduate honors thesis to model my protein of interest, and also how it might be binding to a series of candidate interacting proteins. And as an individual whose scientific research career began after the release of AlphaFold, when I like to reflect on how impactful and widespread it currently is, I think that it's pretty remarkable that something that something that's able to predict with such accuracy wasn't available more than 5 years ago. It truly goes to reflect what an incredible leap forward it is for science, and also simultaneously reflects the incredible amount of work that went into its development. So with that, I ask you to please join me in welcoming Dr. John Jumper for his talk, Highly Accurate Protein Structure Prediction and Its Applications.
That's a lovely introduction. I'm not sure I have any talk left to give, but it was wonderful. It's a real honor to be here. The Gairdner Award is extraordinary, and I'm still in a little bit of disbelief. And I guess, you know, I'm now esteemed. I assume just meant that I got the Gairdner yesterday. But I think it's really exciting to be here and to talk to so many people. And I've already talked to so many people over lunch that have used it or thinking about what the next steps can be. But I want to start with a little bit of audience participation. I just want to know who I'm talking to. So we'll do a little poll. How many in the audience can tell me can draw tyrosine, the molecular structure of tyrosine? Okay, I'd say about a quarter. How many in the audience know layer norm or the formula for layer norm? Okay, just a few. All right, so now I know my audience. So I get to tell you about proteins, with apologies to the biologists and the audience, for whom I hope this is review. And since this is a 30-minute talk, it will definitely be a too-fast primer on protein structure. Proteins are extraordinary machines. Your cell does stuff, and it needs machines to do stuff. And it's found a way to make an enormous variety of machines in an incredibly regular process. So everyone says the DNA are the instructions for life, but what are we building? Right? And there are, in fact, 20,000 regions within our genome, 20,000 genes, regions within our genome that are instruction manuals for proteins, right? And a protein is a very, very simple type of machine. I'm thinking about crossing beyond the two-chair limit, lacking a laser pointer, but nature has come up with this incredible system. So in your DNA, right, you have four choices for each base. But groups of 3 of those, you know, 4 choices, 4 choices, 4 choices, if you do the math, gives you 64 choices, right? And those 64 choices, and you know, map down to about 20 different building blocks. And by building blocks, I mean tyrosine, which I guess, is it on the next? Yeah, it's on the next slide. But you know, these building blocks look like just small molecules that can all be joined together. If you notice, the tops of each of those pictures is the same and the bottoms are different. right? And so the tops are joined together into a chain. I likened it once to, my daughter was, 6, 7, and she has those bracelets that little kids make where you put on one bead at a time to spell something out, and it's the same kind of idea. So the DNA gets converted to RNA. The RNA gets converted into proteins by adding one of these 20 blocks, one after another, which is schematically represented in the bottom left corner as beads on a string, although each of them have a little side chain hanging off. And this is an immensely clever system. Now, you notice that we have a 1D world of genomes. And in fact, we still have a one-dimensional world of these protein chains, right? It's one chain, one after another. But each of those amino acids is a bit different. And my PhD supervisor, Tobin, could tell you lovingly about the individual properties of each amino acid and which ones were disreputable. And, but some of them, for example, are very greasy. They want to avoid water. They will want to clump up to get away from the water, like oil in a pan. Some of them love water. Some of them are positively charged. Some of them can bend both ways. A lot of them can bend only one way. And so we have all these different properties, which means, in fact, that even though we've assembled this line, it will fold up spontaneously into a complex three-dimensional shape just because of the atomic interaction. And these are really, really atomic machines, right? And so the ribosome, a nanoscale factory for building chains, and then those chains assemble themselves. It's like what IKEA would really love to do, right? Right? You take out your chain of bookshelf and then poof, you have it in the correct 3D structure. And this is really important because for a large number of proteins, and certainly not all, only the folded structure is functional. It does the job, It's the things in your muscles that do the pulling, for example. And so if you want to look at these, and you can see kind of your standard chemistry, I can reach this one, ball and stick model, which, even skilled protein biologists probably can't understand much from a ball and stick model, right? They really want to see how this protein chain moves through space. And so Jane Richardson came up with the cartoon or ribbon view, where you can really trace the 3D structure. And this is the exact same protein. In fact, they're overlaid on the other side. But you can see this is an extraordinarily intricate structure. And there are some repeating elements, helices, and these flat things that people call sheets. But the overall 3D packing is extremely intricate, and it's all driven by physics within the cell. And it is extraordinarily important for understanding the function, because you can think about that part of the protein and touching another part, and that's a really important interaction. But it may be very far away in this original 1D sequence, right? You have hundreds of amino acids, and yet the ones that come together. And if you want to understand how this protein does its job, how does it move, how do mutations give us some information about the causes of disease, you really have to understand this 3D structure. And so for 70 years, scientists have gone after 3D structures. Now, the most famous 3D structure is, of course, of DNA, but similar technology. And it's really important to say that we're doing this because we want to understand the cell. And the cell is an extraordinarily packed collection of proteins. I can't remember the exact number, but the cell is only something like 50% water by weight, right? So the cell is, you shouldn't think of this as a lake with two swimmers. You should think of this as, well, being American, a swimming pool on the 4th of July, right? This is an extraordinarily dense, packed mass. This is, in fact, an artist's rendition of a minimal cell, the simplest cell that anyone has made. So 473 different genes in this minimal cell. Humans have 20,000 protein coding genes. And look how complex this is. And look at each of those individual little color splotches are proteins. And often each of these individual little proteins are extraordinarily worthy objects of study that can be a biologist's whole career. There have been Nobel Prizes given for some of the proteins in this picture because they're so important to understanding how the cell functions or fails to function. And so what we really want to do, though, is that this is an enormous, enormous effort in order to take a picture of all these proteins. And taking a picture is hard because atoms are small and light is kind of big, right? So an atom is about an angstrom, 0.1 nanometers. Visible light is about 400 nanometers. So a protein is a couple nanometers in size. So you can't look at a protein in visible light. Well, you can see aggregates, of course, but you can't see the individual proteins. It's just too small. So we need really short wavelength light, one of which is X-rays. So people have come up with a couple of dominant methods to find the structure of proteins. I'll only describe one, where they do 3 magic tricks in a row to get their protein. The first, and probably the hardest, is to convince it to crystallize, to form a regular array, not like it is in the cell, not like it is in solution, but get it to form something that looks like table salt. No one really knows a repeatable recipe to do this, so they try many, many conditions until something works. If you're interested in this thing, I highly recommend the documentary Naturally Obsessed that follows crystallography PhD students who, I don't think I'm spoiling anything to say, they're often not succeeding in achieving this, which is what makes the drama of this documentary. And now, once you've achieved this extraordinarily hard thing, and to get an idea of how hard it might be, is I remember reading one paper, and just in the SI, it said, we did this, and after more than a year, crystals began to form. Which means they tried something, they left it in the fridge, they kept trying things for another year, and then finally found out that thing they did a year ago was working, which allowed them to take their crystals to a rather modestly large facility, like the diamond light source pictured, and to get an idea of the size, that's a car, okay? And this is not like a university building in which there are many experiments. That ring is the experiment. It contains high-energy electrons, I believe it's electron collider, high-energy electrons going around in a ring, and every time you turn a very fast electron with a strong magnet, you can get a blast of X-rays coming off. And so this was an annoying thing for particle physicists, but in fact, this byproduct of particle physics is essential for modern structural biology. And so people have harnessed it and basically built these facilities now for biological and material imaging. So they take these tiny protein crystals. We're trying to look at something extremely small, so of course we go to something the size of a small town. And we blast it with extremely intense X-rays to collect a diffraction pattern so that we can then solve a rather fiendish mathematical problem that doesn't always work, phasing. And if you do all of that, then you get to publish your paper, or at least submit your paper, and you can deposit in the protein data bank, which aggregates this data. And this is extraordinarily difficult. I like to think, and you can do some estimates from some programs that have granted basically only to do crystallography, it's a couple $100,000 on average to do a protein crystal, something of that order, right? And if I tell that to people in industry, they say it's too low. And yet, they're extraordinarily important. You can tell they're important because 200,000 protein crystals, crystals and other structures have been determined and deposited in PDB. So this is a truly extraordinary societal scientific investment in taking pictures of the proteins of the cell. Now, that's great, except geneticists have been even busier. And the sequencing revolution has meant that we know billions of protein sequences, right? It's very cheap to get a protein sequence. In fact, we are learning protein sequences 3,000 times faster than we are learning protein structures, which is not such a favorable ratio. And only a quarter of the human proteome in terms of residues have been covered by an experimental structure with a star for all the details. Now, thankfully, biologists had a lot of foresight, and for the last 50 years, you essentially cannot publish a crystal structure without depositing it in the protein data bank. And this is really, really important. It's one of the cleanest and most wonderful resources in all of biology, data resources. Or if you ask crystallographers, they'll say there's a bunch of junk in there, and you should be very careful what you trust. But from a machine learning point of view, it's extraordinary. And because of that, we have probably the best setup problem for one of the best setup problems for machine learning in all of biology, because we have something we normally don't get, right? A lot of people do machine learning on like image classification, and what's the economic value of that? A penny. Or you do it on text, or what's the value of 10 minutes to write a piece of text? No, we're doing it on a year of work by someone we'll call a doctor at the end, right? An extraordinarily important, valuable problem. And so, of course, I'm talking here because we did well at it. And we've built a system for what I like to cringingly call computational alchemy, where we transmute the base metal of protein sequence into the gold of protein structure. And we did this using neural networks. And this, and to give an idea of the accuracy, on the right is a blind prediction. We didn't know the answer when we made it. Blue is the predicted structure. Green is the experimental structure. So you can see they're extraordinarily close. Here's some more pictures that say, yes, we can keep doing it, right? And you can get some very, very intricate protein structures, including often side chains. If for the structural biologists in the room, we have a new system, AlphaFold 2.3, that was released, I guess, December of last year, that's much better at larger structures. And we started to get into some relatively large proteins, which is good because experimental techniques have moved on to make larger proteins easier to study. And so there's a lot of interest and more data available. But I'll rush through given time. And I want to tell you a little bit about how we did it, subject to the limitations of time. And this is not how you should think about it. This is our network diagram. Each of those blocks actually contains a tremendous amount of complexity, and you can find my talks online where I go into the ideas about some of them. But I think more important is the principle, right? Everyone has the same data in this problem. This isn't a secret data machine learning problem where you get the right data set and then everything falls open. We really needed a set of ideas to do it. Now, one idea was old and been used by basically everyone in the field at the time we started, which is that evolution helps, right? Protein sequence through physics yields a structure and energy landscape, but the protein structure has a strong influence on evolution. Protein structure changes slowly over evolutionary time. You don't want to break this structure. It's often doing a really important function, and you can't break it. And so you can go find lots of related proteins just by looking for similar sequences, knowing nothing about their structure, across human, mouse, dolphin, yeast. And you can get many, many proteins which have not identical structures, but related structures. And so you can say, I had a question, and now I have 1,000 questions that have about the same answer. And that's valuable. That's more information, so it's used as an input. But that alone didn't solve the problem. No one has missed the rise of deep learning. And even when I started giving this talk, 2 years ago, or 2.5 years ago, it was easier to say that. But no one has missed machine learning now. And machine learning has this incredible ability to model lots of things. We model text, we make images, we generate speech. It provides building blocks that are really, really powerful that can do almost arbitrary things. In fact, they're too powerful and generic. They can do anything, so they can't be adopted. They can do anything, so there's nothing that or maybe there's only one thing that they're really specially adapted to. And these were developed in the context of understanding text or understanding images, these kind of core blocks of the network. They don't take advantage of what scientists have determined about proteins. They don't take advantage of this being an atomic system. So like for the first version of AlphaFold we did, AlphaFold 1, basically used very generic computer vision building blocks in order to model proteins, and it kind of worked. It learned some stuff. It was the best system in the world, but it wasn't, I think, good enough that biologists should really base months of experiment on what it said. And so what we did is we went back to the drawing board and we said, how are we going to really think about this problem anew? How are we going to take our physical and geometric insights about this problem and build it into the network structure? And I like to think of it as we were doing the science of doing the AI of protein structure. We are going to build our own machine learning for the problems, the situations, using the intuitions of protein structure. And the core team was maybe 15, 18 people who started out, I have a bit of bio training, but I'm really a physicist. We had one person who was a proper biologist, but mostly we had physicists and computer scientists. But all kind of jumping in, understanding biology, and trying to figure out what is the right expression into neural networks. For example, how do we write down the right set of mathematics that we can produce structure directly instead of making predictions about the structure? So we can immediately be wrong, and we can be wrong sometimes at multiple proteins per second. And if you can be wrong really, really fast, then you can try lots of ideas to fix it, right? Also thinking about how do we think less about the protein sequence? We knew that at the beginning. How do we think more about the spatial structure? How do we think about when two parts of the protein come together? How do we make sure it knows that these two parts are near each other? How can we establish very fast information transfer across that, rapid communication among spatially close residues? How will it reason under uncertainty? How is this kind of uncertainty done? And given the time limits of this talk, I won't tell you much about how we actually do it. But what I will say is that it really, really mattered. So one external study by the Al-Qureshi lab said, well, what would happen if we take the AlphaFold 2 architecture and train it on much less data? When would it be less accurate, this specially adapted one, be less accurate than AlphaFold 1, which is a traditional architecture trained on all the data? And they found that it was about 1% of the data. So if you train AlphaFold 2 on 1% of the PDB, it's as accurate as AlphaFold 1 trained on the whole PDB. So AlphaFold 2 is 100 times more efficient at using this data, and that's just a matter of respect. These are extraordinarily precious data. We need to treat them, we need to use very, very efficient machine learning in order to get the maximal accuracy, because it's very, very hard and expensive to increase that data. I'll skip ahead a little bit and say, as we built this, or after we built this, we also said, well, how does it work, and can we get an insight? So we realized we have a layer-wise structure, and we thought that it might be doing something iterative and refining, and so we could simply train new little pieces of AlphaFold, hold part of it fixed, and say, what's your answer after the first layer, the second layer? There's about 192 layers here. And what we found is we could actually get a pretty good picture of the trajectory of AlphaFold solving a structure. This is a large RNA polymerase. On the left is a comparison of the final answer accuracy. On the right, and you can see a layer number in the bottom corner, is every step of the prediction predicted as an individual response. And you can see an iterative refinement. If you look closely, you can see some parts of the structure, for example, the top right, are solved almost immediately. There's very strong, say, evolutionary information or other information that makes it a very easy part of the structure to solve. It's like a jigsaw puzzle where the corner is easy to find. But then if you look, say, at the bottom left, you can see this. I did it. You can see this part of the structure in the bottom left actually takes nearly the whole depth of the network to solve, and really only is able to solve it when it has solidified the rest of the pieces and is able to figure out how that piece attaches to the rest of it. So you have this iterative refinement, and there are many other kind of ideas and processes, but we built this iterative refinement so that the core of AlphaFold, the part that makes it really more accurate than what came before, is it can do extremely precise refinement of predictions to really, really high precision structures. Now the second thing is, if we built this and we said, here's an answer, and sometimes it's great and sometimes it's terrible, and even worse, if it hallucinated a bunch and it made some really beautiful things that were sometimes entirely wrong, it would be very hard to use. But what we realized is kind of, well, one, we got lucky, which is that when AlphaFold doesn't know, it tends to produce something very ugly. And if you see the red bits, no structural biologist is going to think that looks like protein structure. That looks like what people have taken to call spaghetti. And so when AlphaFold is wrong, it generally produces an ugly answer. The second is that we were able to do a very simple procedure, like how do you figure out if these structures should be trusted? We decided to just ask AlphaFold. Or more specifically, during training, as we would produce an answer, we would also have it produce a confidence, and we would score it on how well it predicted its own error. And I like to liken this to coming out of a test, and someone asks, how did you do? And you said, I did really well in question 1, but question 2, I'm pretty sure I missed it, and question 3, maybe. Right? You don't know the answer to question 2 or question 3, but you know that you probably didn't get it or you probably did. AlphaFold can make a very similar judgment. We just project this. And there's some details to it, but this works quite well. And so these have proven to be quite robust measures. So rather than try and build, say, interpretable AI, where you trust it because you believe that you could have done that algorithm, we're building trustworthy AI in a different way, that it has a high chance. You can measure its calibration and knowing when it's wrong. So you can have an estimate of how trustworthy this is, and you can prove this estimate works across general classes as well as your own problem class. Now, we built a bunch more ideas. It's a 30-minute talk. Which parts of it mattered? What was the, people ask, what's the one idea that converted AlphaFold 1 into AlphaFold 2? And the answer is there isn't one. There's probably 6 or 7 or 8 ideas. This is showing the delta if we basically do knockouts on ideas or remove an idea, ablations for machine learners. If we remove an idea at a time, how much less accurate is the AlphaFold system? And you can see all of them are a bit less accurate, but AlphaFold 2 is about 30 points more accurate than AlphaFold 1 in the scale on the left. So none of these ideas are worth that much. They all stack together. We needed many ideas, each of which increased our data efficiency a little bit to get to this final system. Now, when you start making out multiple ideas, like the bottom row, no IPA and no recycling, then you get some larger effects. Double knockouts are worth more than singles. But still, a lot of it was stacking many good ideas together and working very efficiently to achieve it. Now, quickly, and how much time do I have? I don't know. I'll do some results and applications or talk a little bit. The one thing to say is that you should be extraordinarily skeptical of claims in machine learning. Everyone in machine learning is, you might as well too. And how do you really know? And in fact, there were claims really before this was considered a machine learning field, but protein structure prediction. In 1994, there are claims that protein structure prediction is essentially solved. And so a couple very good scientists, including John Molt and Christophe Fidelis, says, oh, well, if it's solved, you wouldn't mind if I checked you on that. And I have, I'll contact some of my crystallography friends and ask if they will tell me a protein that they have determined the structure of, but they haven't published it yet. And I'll send that to you, and you send me back the structure, and then we can all agree it's solved. And the first time so they called this the critical assessment of protein structure prediction, where the P got lost in the acronym and is pronounced CASP. And the first time they ran it, it was a clear disaster for the protein structure prediction field, that it was very clear that we could not predict protein structure. And it was only because they did this very rigorous blind assessment they were able to know. And they continued doing this assessment every 2 years since 1994, so we're now in the 30th year or so of CASP. And of course, finally in 2020, we produced a system that was very accurate, sub-angstrom accuracy, about a third of the error of any other system in that CASP. And really, this is how we know it works, right? You can only really know it works when you're not, there's always a tendency in computational systems that when it doesn't work on a problem, you change your ideas, and then it works on that problem, and then you say, look, it works on this problem. But that was your training set. That's what you use to build the ideas. You need really careful, blind, rigorous testing. Now, we've made it very widely available in two ways. We have open sourced the software. You can download and run it. There's nice Colab interfaces. Colab Fold is very nice if you want to get a structure prediction. For single structures, in collaboration with Imble EBI, we said, well, AlphaFold is pretty fast. What if we run everything? In this case, all of Trimble, or all of Uniprot, almost. And so we have released a database of 200 million predictions, 1,000 times larger than PDB. Now, these are all predictions. Some of them have no confidence whatsoever, but it's a resource, and everyone can go click and use it. And it's a starting point, but you can also run the software, and you can do new types of investigations. And people have done things like hunting for new protein folds or other large-scale investigations into this. Now, one thing I'll take an aside to say is that any protein biologist will tell you there's a lot of context to proteins. How many copies, whether there are ligands, does it stick to DNA, what pH should it be at, what conformation. And AlphaFold is relatively tolerant to not knowing this. In fact, we don't give it away. We can't tell it pH at all. And we can't, and the original AlphaFold, a later version AlphaFold Multimeter, you can't even tell it how many copies. And this is a trimer, 3 different copies of the protein that twist around each other. And you can see that AlphaFold produced an extraordinarily good prediction of this, even though it makes no sense as a protein structure in isolation. You need those other 2 chains, which are grayed out. And what AlphaFold is really saying is not, this is the structure in isolation, this is the structure in a context where it might appear in PDB. That's a superpower if you want to do 200 million predictions and you have no way of getting 200 million experimental conditions right. It is a challenge if you want to say, well, how would it be different if I changed oligomer state? Well, in that case, you can control it with multimer, but pH or other things. And that's an area, I think, for future work. It's probably part of the reason that AlphaFold is less mutation sensitive than we would love. We also found after we released AlphaFold that people did creative stuff with it. They used it off-label. And what we would find is, like, one of the big areas was they would find protein interactions by simply taking a big collection of proteins and saying, which ones can AlphaFold produce a structure sticking them together? And sometimes this is done in collaboration with experimental data, where you have a list of possible hits. And sometimes this is done simply on a smaller set of proteins all on its own. And people have discovered lots of novel interactions. In fact, some recent science papers that run AlphaFold on, make 70 AlphaFold predictions, find the 6 that stick to this protein, make a model of how this is involved in DNA regulation. And it was essentially, I think, confirmed in some independent work that actually had a structure. So there's a lot of work in interaction discovery with AlphaFold, which was somewhat unexpected. I'll skip some of the protein design stuff. But I think maybe one story I really like, this story of midnolin. This protein that was discovered by biochemical means recently. Basically, it was uncharacterized almost. And then it was discovered to be essential for degrading proteins. For people who don't know, your cell has lots of mechanisms to get rid of proteins. And it has a machine that chews up proteins, the proteasome, which is protein, that will chew up other proteins, but then you have a whole set of things to mark things to be killed or to drag it over to this proteasome so it can be destroyed, basically taking it to the shredder. And one that wasn't known was midnelin. And there are, in fact, hundreds of genes that are involved in a certain phase of the cell cycle. And then the cell needs to get rid of them. And they found through basically random CRISPR knockouts that this protein midnelin, if you knocked it down, then these proteins would stay around. So this was involved in degradation. And then they turned around and said, well, if I add more midnelin, how many proteins are gotten rid of? And it's about 500. And they ran them all through AlphaFold, right? To say, can we understand how they're degraded? In fact, they got a really, really clear signal. Oh, I've got a cursor, yeah. That this is the pink and the yellow or the midnelin protein. The red is another protein. And in the presence of that protein, AlphaFold predicts that midnelin forms this 5-strand sheet. Basically, they named it cleverly catch 1 and catch 2, and the target is in between those two. And of course, this is just a prediction. How do they know if it's right? Well, they did the obvious biochemical thing. They made a version of this protein, or for beta, without this strand. And then suddenly it doesn't get degraded. Midnelin doesn't recognize it. And in fact, of 200 proteins where AlphaFold was able to bind it against midnelin, produce quite similar structures for almost all of them. And then for 5 or 10 of them, they tested deleting the exact region that was predicted to bind midnelin from those proteins, and that abolished their degradation, except in one case where AlphaFold predicted 2 regions that bound midnelin. And if you delete one of them, you get a reduction in degradation, but still substantial. And if you delete both, you get full reduction. So they've started to work out, really, and I think what's exciting about this is it's a really great paper anyway for the molecular biology, the biochemistry. But we get to skip a bunch of steps and go straight to mechanism by using kind of AlphaFold in this playful way, making hypotheses, testing them, and using it in conversation with experiment that would be done with structural biology, but it would take classical structural biology, but it would take a lot longer and be very, very difficult, and people may not know where to start. And it's this way in which AI is starting to accelerate science as part of the larger science ecosystem. And it's not really that AlphaFold is really important because you press it once and you get an answer and it turns into a pill for cancer. It's because it helps scientists do some of the really hard parts of the incredible biological scientific enterprise. And it helps them do it faster. And because it's orders and orders and orders of magnitude faster than experimental structural biology, people can try new ideas and become more playful with it. Now, finally, I want to talk to you about, I told you I was sad before that AlphaFold isn't too responsive to small changes in the protein called mutations. And so we've done some work on predicting whether a mutation, in this case, is harmful in a genomic context or is uncommon in the population. This is the alpha missense. And a large number of human possible variants, things that might come up in a genetic screen, especially a trio, have unknown disease implications. And we can show that by basically training a version of AlphaFold, right? Structure should be useful in thinking about whether a missense mutation is harmful or benign. And by training a special version of AlphaFold to predict, in essence, is this a common, it actually has quite a bit more accuracy than earlier systems at confidently saying whether this is a likely or unlikely mutation, which is a step that can be used in, or hasn't been used yet because the paper just came out, but is a step that can feed into clinical diagnostic pipelines and thinking about, of the mutations that a patient may have, which do we think is actually the harmful one, especially for congenital disease. And so it's kind of ongoing work, but it's also showing how, like having a sense of structure, how do we bring it to other problems like genomics where it helps but is not definitive? And this is kind of one paradigm for doing it. Finally, I want to close and say a little bit about organizing research for machine learning. What should we think about? And I think one of the big lessons of recent machine learning is that really big data helps. And really, really diverse data is even more important than really big data. So the PDB contains the full output of the structural biology community. So when you train machine learning to do well on it, you train it to do well on anything that might appear in your training set, which is pretty much anything that might be produced by a structural biologist of a protein structure. If you had much narrower data sets, like a specific family of proteins that was very large and you trained AlphaFold on it, it may only learn to do that family. It probably couldn't do others. You really need diversity in your data much more than size. You know, I can't tell you how many people say, you know, I have a petabyte of data. You know, it's all images of one type of surface in a TEM. And I'm like, well, that's, you can learn a machine learning model, but that'll be a model of that particular surface, not all surfaces. And bigger problems are easier to work with ML. This has been one of the, or really there's two classes, but the deep learning, what we would call AI, is much easier on much larger things, can learn much cleverer facts rather than small targeted data. And so if you wanted the structure of a specific protein, the best way we would know to do it is learn the structure of all proteins and then apply that system to the one you're asking about. It really matters also doing the science of machine learning, really thinking about there isn't one AI and you apply the AI to your problem and it's all driven by your data, right? Even when people say it's all transformers, when you dig into it, it's actually not all transformers. It's an important gadget, but it's only a part. And doing this right can be worth orders of magnitude more data. And it's a lot more fun than gathering data. And this really mattered for AlphaFold. This was the difference. And finally, measure really, really, really well. For one thing, you know, I believe firmly that you should only time split. So when we developed AlphaFold, we said April 2018. Everything before that is allowed for training. Everything after that we will only use to check how good our system is and make decision on system architecture. And we were very, very strict on that because we were going to train our machine learning systems in the past and we're going to use them in the future. That's the way time works. And so it's really important that you test with that kind of temporal split, that you're very careful on which examples are truly novel and which are very similar to ones that have been solved before. And if you're an experimentalist and says, how can I contribute to machine learning? I tell them, don't worry about making a training set. People can find those. Make an incredible evaluation set. An AlphaFold was built 1 or 2% improvements at a time, so your set needs to be much larger than you think so that you can make an extremely low noise measurement of what helps, right? You can't stack 1% gains with 10% noise. And so you really have to think a lot about evaluation, and doing evaluation really well is the difference between top-tier machine learning and machine learning that doesn't work. Finally, like how should we think about this going forward? And we all very much believe and hope that AlphaFold will be, you know, maybe hopefully looked at as a milestone, but as like opening the door to AI in biology, AI for science, that there are many more problems in which we can amplify the incredible work of experimentalists, that we can take data, diverse data collected across many problems with an underlying physics, but still very sparse and not the one you need to do your study up in the top right corner, and fill out that picture with machine learning and use that as a stepping stone to do science faster and more creatively. And with that, I want to thank everyone who made AlphaFold possible, the experimentalists who deposited data, the team at DeepMind that worked on it, our wonderful collaborators at MBI, the CASP community, and you all for listening. Thank you.
So we have time for a few questions. We might have time at the end for some more. If you have questions, there are mics up here. If you could please come because it is recorded. We would like to capture the question.
I can repeat the first one too if it helps.
Talk a little bit about what you mean by how AlphaFold is able to find new folds.
So the question was, how can I talk about how AlphaFold can find new folds? One that we did a careful assessment of is that, so new fold is a very kind of technical term. It doesn't mean structure. It means a particular arrangement of secondary structure elements. What we found, and people often, you know, old systems would do fold recognition and say, I know that fold, and then try and build the chain into it. And AlphaFold just really doesn't do that. It works much more locally. So we show no reduced performance on novel folds. And I think it's really because fold is something of a human concept, right? That this kind of pattern, there's a couple thousand known folds, but I think it's probably just, my quiet personal belief, you know, recorded for posterity, is that I believe that folds are, often they're useful, but they're a little bit too human. They have some of the problems that, for example, cell types have, where when you get into it, there's so many exceptions and other things.
It's a bit counterintuitive, the idea that there's a training set and then you find things that I guess there might be things outside of it.
So you could think of it this way. The training set is so diverse that it's not memorizing the training set. It's learning an algorithm that worked on the training set. And if that training set is diverse enough, if it's very not diverse, then it memorizes all the answers because it has, enough data that it can just store the PDB. That's not a problem. But if it's really diverse, it actually ends up learning a very general algorithm that happens to work on the training set. And so I think that's what we're seeing.
Excellent talk and quite inspiring, I might say. You touched on the fact here.
Thank you.
You touched on the fact that when it came to the AlphaFold, either the 1 or the 2, parameters such as pH or I'm assuming even ionic strengths weren't really involved. Do you think that looking into the future, because many of the structures, as much as we love it, and I do, were actually determined outside so-called,
They were
native
very
conditions.
outside physiological conditions.
And I did appreciate the fact that you showed the cell as being very crowded. I mean, teaching biochemistry and sometimes simple systems such as microorganisms, we have to tell the students that, it's not like, moving freely. It's quite crowded out there. But that does impact folding as well. So moving forward, how do you see these other elements improving or getting more closely to what a protein may look like in the cell rather than in a petri dish?
I think there are 2 or 3 things. One actually does work in our favor is we're trying to predict the structure in PDB, but we are worse at predicting crystal packing artifacts than other parts. In a sense, they're driven by a different physics or they're not driven by evolution, certainly. They're not evolutionarily encoded. So we will often find differences against the crystal, especially in crystal contact locations, which might mean that sometimes we're right. I think on the larger question of how do we get all these modulators, I kind of personally believe it's wrong to think of them as changing structure. They change energy. Or in a sense, they raise or lower the energy of given structures, and you may have a phase transition. But because of that, I think to really think about these, we're going to have to think about delta Gs, delta Gs, whereas AlphaFold is trying to build kind of in a geometric, non-energetic picture. I mean, people have found a bit of energy. So I think we'll really have to start thinking about stability, energetics, delta G, delta G, and then we can think about ionic strength and others. That's my personal theory.
Thank you.
Okay, would you encourage me to use AlphaFold to determine protein-protein interactions if I have the structure of one and I have ideas about potential others? I mean, this could cut down, my trainees' work by many, many months, if not years.
So AlphaFold multimer is specifically trained to produce interactions. It works on something like 2/3 of PDB cases. And the confidence is pretty good, so if it's confident about the interaction. And there are tricks that people have used. So some are known not to work well, like antibody-antigen binding doesn't seem to work very well at all. It's not because it's evolutionarily encoded, I think. It's just a hard problem. But even there, like Bjorn Wallner has shown that if you run AlphaFold a whole bunch, and by a whole bunch, I mean he ran it like 60,000 times on one target, that it can get lucky, but when it gets lucky, it knows it. And so if he takes the most confident structure, he's was able to produce some very good predictions in Cas15 using this, using the strategy of run AlphaFold a whole bunch. But yeah, I would encourage you to try it. If there's one structure known, you can often use it as a template. Now you want to be a little bit cautious because you know it in an apo form, at least relative to that protein, so it may move. I'd probably try it just out of the box first or without templates and kind of play with it. I think what people have found, the intuition has been if you can get AlphaFold to be confident about something, it's probably a meaningful state. It may not be the state in your conditions, but you have a high chance in most cases. And I think the alternative is just not trying it all and doing months of experiments. You might as well spend 2 days trying to make AlphaFold work, and you can always do the experiment afterwards. I don't know.
Okay, I think, Brittany, would you introduce our next speaker, please?
So it's now my pleasure to introduce our second speaker, an esteemed physician and scientist whose research is focused on developing evidence-based practices for critical care medicine, Dr. Deborah Cook. Dr. Cook completed her medical doctorate at McMaster University and her fellowship training in critical care medicine at Stanford. She then returned to Hamilton as a clinically practicing physician, researcher, and professor through St. Joseph's Hospital. Dr. Cook is a distinguished professor of the Departments of Medicine and of Health Research Methods, Evidence, and Impact of McMaster University. She is a fellow at the Canadian Academy of Health Sciences, a fellow of the Royal Society of Canada, and officer of the Order of Canada. Dr. Cook is the recipient of the 2022 Canada Gairdner Award for Outstanding Science and Leadership. Her work has had a significant impact on the treatment and outcomes of intensive care patients on a global scale. Her research involves developing best practices to prevent ICU-acquired complications, infections, and disease. And these care practices have been internationally adopted and have been shown to reduce morbidity associated with critical care complications. Dr. Cook's research also involves assessing and developing best practices in compassionate end-of-life care. And part of this research founded the 3 Wishes Project, which aims to better support dying patients by honoring their lives, easing family grief, and fostering humanism in practice. This project has also seen global adaptation and supports the significant impact that Dr. Cook's work and leadership in this community has had on patients around the world. So in her 30 years of work has impacted individuals directly through her own clinical practice and indirectly through the positive policy changes that have been implemented because of her research. So please join me in welcoming Dr. Deborah Cook for her talk, Critical Care Research from Humble Beginnings to Global Reach.
Thank you. Good afternoon, everybody. I've learned so much from John already, and I just can't wait to discuss this with people who don't know anything about it, and just try to just share a little bit of the knowledge, modest as it is, that I've now acquired thanks to you and thanks to the Gairdner. This is a story about critical illness and the clinical research that starts with patients and ends with patients. And just for fun, I thought I'd show some pictures of the women who I have had the pleasure of learning from, teaching, working with, collaborating with. These are just the women in Hamilton, which is my academic home. My 2 mentors were Dr. David Sackett, one of the founders of evidence-based medicine, and Dr. Gordon Guyatt, another founder of evidence-based medicine, who coined the term. They were both visionary epidemiologists and internists who wanted to help practitioners read research reports and, when suitable, critically appraise them when they could, and consider them in the care of patients. And of course, the research that we read as clinicians is more clinically based than basic science-oriented or translational. But it is built on, clinical research is built on the genius work of others doing laboratory-based work like John. And these 2 mentors were absolutely incredible. There are other people that you've all had in your life. You have mentors, I know, but there's so many different types of mentors. And then there's people who aren't really your mentors necessarily, but they're just people who are out there, people who you look up to. Literally, in the case of Roberta Bondar, she's from the University of Toronto originally, I think, in family medicine. Oh, that was May Cohen, I'm sorry, is from the University of Toronto. But Roberta Bondar was Canada's first female astronaut, and really strong scientist. And she did her medicine, I believe, at McMaster. And May Cohen, who was a very strong female scientist, and I just wanted to acknowledge all of the people, and I hope if you're taking a moment to reflect on those who've helped you or inspired you, you'll think about some of the informal mentors or just colleagues who are out there. They might not be specific allies or supporters in any way, but they make a difference. Welcome to my world. It's the world of the intensive care unit. Replete with technology. You can probably recognize some oxygen device and tubes, oxygen, blood coming in and out of patient dialysis machine. What you'll notice in the intensive care unit, the modern day intensive care unit, that it's really hard to identify, is somebody in the bed? Yes. Hard to say who that person is. And this is, in fact, a very dehumanizing place where we speak often about the biology of the person. We know their lab data. We know their tests, other tests, radiologic tests. We might know they're admitting problems. And we certainly know about all their complications. But it is a place where we have more questions than answers, even to this day. And in Canada, in the early days, 30 years ago, when I was doing my training in the States, obviously a huge population, many more patients, and lots of research opportunities. There's great interest for everyone graduating to work in the lab. And I was interested to be a little bit more closely aligned with patients. And so we decided in Canada, the group of us, that we would actually try to collaborate. And because it's a relatively small country in terms of population, but big in terms of geography, we knew that we could achieve more together than we could as individuals. We wanted to have larger studies. At the time, the average sample size of some of our randomized trials was in the 20s or 30s, and it's really hard to make inferences from small studies, as we all know. So we wanted to collaborate coast to coast and actually share our skills. This was quite a novel concept, the antithesis of what we've heard about today from John. People did not share their knowledge. Even if they're going to collaborate, people kept a lot of their wisdom and their ideas rather close to the chest. And we knew we wanted to do things differently, create a nurturing venue, and enjoy the inimitable Canadian camaraderie. And we had this mission, and we didn't need a big, huge, fancy company to come together to say, what is your mission? What are your values? But we said we wanted to try to advance the care of patients and improve the outcomes for just children, critically ill children, of which mercifully there are fewer than adults, but children and adults who are seriously ill might need life support through excellence in clinical research, but keep it very clinically grounded. And we further went on to be explicit about what we valued in this case when we come together as a group. And it was obviously about the scientific creativity and the integrity, but we wanted to stay clinically relevant and have our questions arise from the bedside, which was also a rather unusual feature at that time, and ensure that we're always collaborative. We had a sort of saying in the early days for this Canadian Critical Care Trials Group that I was very grateful to sort of co-found and be a chair of for 8 years. We said, check your ego at the door. You're welcome here to be open-minded, share your ideas. If you're going to critique, do so constructively, and let's help each other make our science better. And we have the strong sense of responsibility because in Canada, obviously, research funding is precious and minimal, and we wanted to make sure we did as much as we could with the resources that we had. We also thought about organizational culture, which itself was sort of novel. And the way I think about organizational culture is if you imagine how personality is to an individual, organizational culture is to a group. So there are different ways to view organizational culture, and all of you are part of many, many organizations, formal or informal. But there's approaches, and we thought we want to be developmental, we want to be innovative and rational, but we don't want anything hierarchical, and we got together very much in a group. So when humble beginnings is what I'm talking about, where we had nothing, we had no research group, we had no collaboration, we had no money, but we had a lot of ideas. And this, in many ways, ironically, I think was part of the genesis of the Canadian Critical Care Trials Group, which is the first consortium for critical care clinical research in the world. It's been emulated everywhere. And our capacity has gone on, and here are some of the chairs. Our first chair was a thoracic surgeon, an unlikely figure to be, unlikely maybe background to be chair of the Canadian Critical Care Trials Group, but he was very, very wise and very egalitarian and super helpful. Myself, Dr. John Marshall here at U of T, Paul Hebert, who's at Ottawa, Rob Fowler, who's helped us hugely through the pandemic, along with John. He's at Sunnybrook. And now Kusa Menem, our first female and pediatric critical care intensivist. So the organizational culture was super perpetuated by all these folks. When we started, we thought, okay, we've got ideas, we have no money, but we've got a will, we've got our mission, our vision, our value. What are we going to do? And we knew that our connection was very, very important, and this was very much in person at the time. And we got together to design studies as a group. Again, very different than one person thinking for days and days and hours and weeks and doing a few projects. We collectively designed our studies and had great discussions to try to sort of filter which would be the studies that were most likely to have a big impact. And of course, they had to be rigorous, they had to be relevant, they had to be ethical, they had to be feasible, and they had to be fundable. And it's rare to have studies where all of those features exist. So it's very hard for that. And I spoke a little bit this morning about our gastropulmonary research program, and this was the study we were going to do right at the beginning, was right in the middle, a randomized clinical trial comparing two different approaches to try to prevent bleeding from the stomach, stress-associated bleeding from the stomach. And we thought, okay, we're ready to go. Let's do this randomized trial using the two different drugs most commonly employed to prevent stress bleeding from the stomach. But we realized quickly in a programmatic approach that we didn't know enough to do a trial right away. So we did a pilot prognosis study to just enroll, there's actually only 100 patients, to see what happens to these people who bleed. Do they do well? Do they need a lot of resources? Do they die? And we did a bigger multi-center bleeding and prognosis risk factor study across Canada. And then, as you know, whenever you're going to prevent something, you have to ask, who cares about it, really? Like, how important is it, this thing that you're going to prevent? Everyone understands now with COVID, obviously, prevent it with vaccines. But there are many things in medicine. Okay, rabies, yes. Tetanus, yes. People get it with vaccines. But sometimes when they're critically ill patients and there's a bunch of little problems, people wonder, should we bother trying to prevent this bleeding? So we did a case control study to show, yes, there's an attributable morbidity and mortality, and actually there's cost, there's healthcare resource consumption. And then, as you all know, nowadays doing a meta-analysis, systematic review to find out which are the two best ways to prevent bleeding. So there's this whole series of studies before we went right to the randomized clinical trial. And in retrospect, this was very, very smart, even though people thought you're not going fast enough. And the pressure today is always to accelerate your research. But we took the approach of slow down and figure a few things out. So had we not done these studies, we wouldn't even have a proper definition of bleeding or understand what criteria would be necessary to define someone as having clinically important bleeding. We wouldn't know the proper estimate or the incidence we're trying to prevent. So we couldn't make a sample size calculation. We had to imagine getting funding. Is this important? Making a case. So all of these things were averted, and then we were able to responsibly use our resources and do that trial. Here's the finding. The antacid ranitidine was associated with a 50% lower bleeding rate than the sucralfate, which is a weak base, sort of a fluid that people ingest to this day to sort of bathe the lining of the stomach and mitigate the risk of ulceration. So you can see the bleeding rate is very low overall, but there was a 50% reduction, and then antacids became the mainstay of prevention, and still is the case for 30 years. But there were some side effects of worry. Ventilator-associated pneumonia. You could say this seems like there might be an increase in risk of pneumonia, but it didn't pan out statistically speaking, and as we expected, there's no difference in mortality. So our whole approach to one study, when you collaborate widely, was not to do one study at all, to do the preparatory work, one randomized trial, and with that database in a way not nearly as sophisticated as what John has described. There's no AI at that time, but we did use the database to ask questions of it, not to torture the data, but to try to understand, given the limitations of this database, rigorously developed for the purposes of a randomized trial, what else can we learn? And you can see we repeated the case control study to understand the attributable morbidity and mortality again, as he pointed out. Do it once, test it again. Is it still true in a different database? And went on to study ventilator-associated pneumonia. So this taught us early on that if you are working with a group of people and you have a longer-term view, you actually can have the opportunity to answer and ask in the first place numerous scientific questions and then improve the methodology along the way. And this was sort of Canada's story, and as people like to point out from other countries, well, you don't have any money, so you have to take it slow and do it properly. And we say, yes, thank you. We did it very properly. So when we think about collaborative research that's clinical and patient-based, we know it's crucial to synergize and energize from each other. And our notion at the time, after working at it for 10 years or so, shifted a little bit more to mobilization of that evidence to be sure that we were able to put it in the hands of clinicians, nurses, doctors, respiratory therapists, physios, and others, to actually apply it to patients. So we started to think about knowledge mobilization. We focused on knowledge generation. A lot of science is about knowledge generation. But also there's a whole aspect of science which is knowledge translation. And I wanted to share just another example about blood clots to sort of illustrate what was at the time pretty unique in the, I guess, soup to nuts approach from knowledge generation to translation. So many of you would be familiar with thrombosis or blood clots. One of the major risk factors for blood clots is immobilization. People who take long train, plane rides, etc. Malignancy, birth control pill, other things. But critically ill patients obviously are immobilized, sedated often, paralyzed sometimes, nearly dead, high risk of mortality. And so they certainly have stasis of blood flow. They have vessel wall injury because we're poking them all the time, catheters everywhere in the blood vessels and elsewhere. And they're hypercoagulable, so hypercoagulable state is part of being critically ill. And so you can imagine people who are critically ill are at risk of blood clots. And when we were doing an environmental scan to see, okay, let's look through all of the big guideline documents, white papers, consensus statements to see what's being said about critically ill patients, they weren't even mentioned in these documents. So this is another example of doing an environmental scan and going where the holes are. Trauma patients who are also critically ill after MVAs, etc., they were mentioned because they often have long bone fractures that puts them at risk of blood clots. But other sort of post-op patients and folks from the community coming into the unit weren't even mentioned. So we knew we were in luck for another field with a big hole. And there is two kinds of heparin I mentioned this morning. Unfractionated heparin, which is an old, very widely available, very safe, very effective drug that helps to prevent blood clots. And then there's the low molecular weight heparin, newer type of heparin, more expensive, more effective in many different trials. And we thought, let's start as another way to begin your science. You know, when people, it's this way, no, it's this way, this is better. Again, it's great to be able to document that. And so we did a survey, a self-administered survey, no e-survey at that time, paper-based, of ICU directors across France and across Canada. What do you use in your ICU when you're critically ill patients? And all of the Canadians were saying, of course, the unfractionated heparin. It's less expensive, very widely used everywhere. It's perfectly fine. And people in France were saying, no, we use the new low molecular weight heparin drug. And there's other approaches, mechanical stockings and pneumatic compression devices. But again, this is a survey as a second methodology to help set up your case. It's like, wow, we got confusion. People are doing exactly the opposite things based on the evidence available. So you know you're in another good zone. We designed a study, a simple comparison of low molecular heparin versus unfractionated. And we included, in this case, we were focusing on adults, in all those people who we would otherwise be giving one of the heparins, excluded people who should have low molecular weight heparin, that's the trauma and orthopedic population, and people who should not have any heparin, of course, and people who must have high-dose heparin, and went ahead to do this big RCT in several centers in all over the world. And again, just to show the program of approach, the preparatory work that we did, bunch of surveys, observational studies, et cetera, and PROTECT, which is the name of the trial, is one piece of this research program. So this collaborative model, which has the long view, which is one way to look at your life, if you're fortunate enough to have a long life, it's not about the everyday little ups and downs and all the barriers that they're road bumps. But having the long view, this is the kind of thing that is possible with a big group of people. And we went on to do more. The results I just wanted to share with you, the low molecular weight heparin was associated with slightly less DVT, or deep vein thrombosis of the leg. That's there in red, but not much compared with unfractionated heparin. You can see that the hazard ratio there, if it's 1, there's no difference between these two drugs. Slightly less than 1, you can tell this is not statistically significant. So there's really no big difference when it comes to blood clots, which is the primary outcome. More serious kind of clots, blood clots from the leg, can go to the lung, as you probably know. And there was a 50% reduction in lung clots with the low molecular weight heparin. So that was important. And if you think HIT is a heparin-induced thrombocytopenia, which is a potentially fatal drug allergy, that was also 50% less likely to occur with the low molecular weight heparin. And importantly, no difference in bleeding risk. That's the hazard ratio of 1. So depending on if you're a trialist and you're focusing on the primary outcome, you say, well, there's no benefit to low molecular weight heparin. It did not prevent statistically significantly blood clots in the leg. True. But if you're a patient, you'd say, wow, everything that's bad that could happen to you associated with this condition is either just as likely to happen, a little bit less likely, or significantly less likely to happen. So patient and family perspective, this would be a trial where you'd want to shift everyone over to low molecular weight heparin if the citizens of the country were in charge. But they are not, as you know, in charge of drug prescribing or purchasing drugs. And the other thing in clinical research we look at a lot is the number of events. And you can see when there are very few events in question, the data are a little more unstable. We can't be sure. Few patients going one side to the other, the results might have been different. But overall, this was an extremely instructive trial. And the catch of the low molecular weight heparin being more expensive really caught people, because then what do you do? Primary trial did not achieve statistical significance on the primary outcome, but everything bad for you is less likely to occur or with similar frequency for the patients and families, but the drug costs more. So what does the healthcare system do? So with the help of Rob Fowler and others, we did an economic evaluation and showed that it is cost effective to use the more expensive low molecular weight heparin, which is just a few dollars really, but because of all the bad events that are being prevented by using a better prophylactic agent, that's the thing that saves money. And we're able to sort of put this into a metric. If an ICU with 1,000 medical surgical patients get admitted over a year, and you use the less expensive heparin, because you're going to have more bad outcomes, you're going to actually incur costs. So it's an example of you're wasting money when you use what you think is less expensive. So it was a great example of thinking about our healthcare system. And the last example I wanted to review is called the level of care study. I was mentioning this morning with the students who seem to have an acute awareness, having gone through the pandemic, of the fatality associated with critical illness, obviously pre-pandemic and forevermore. But Canadians, although they say they'd like to die at home, it's not a reality for many people, socially speaking, economically speaking, just from a feasibility perspective. And while we would believe in most of the parts of the world, not just the so-called Western world, that a good death is a basic human right. That's far from the reality in the world today. And we know in the ICU that even though a lot of patients do die, with the dehumanizing sort of world in which the ICU can be, it can be extremely stressful. So many years ago, I was interested in studying dying in the ICU, and I was sort of advised by some of the senior folks around in my life at the time that was really not science. That's just too soft a topic for a senior scientist or properly trained scientist like me, and it wasn't, wasn't a good avenue to pursue. And so I kept it as a favorite part of my practice and a special sort of pet topic I enjoyed teaching on. And I didn't really pursue it too much, but I knew there were a lot of knowledge gaps in the understanding the way people died and what family preferences were, et cetera. And obviously, as has come to light, you will all understand the light being shone on the moral distress around death and dying in the ICU recently. And so we thought we'd better try some sort of research. And what we did is picked an approach that might be viewed as a low-hanging fruit, but another way to look at it is the only admissible study design we could pursue was an observational study at that time. We didn't know enough to intervene. We wouldn't have been brave enough to do so, because we wouldn't know what to who to intervene on and what to use to intervene. But we observed patients in several countries, and I'm not going to tell you all the details, but it was in Sweden, Australia, and U.S. and Canada, and we just enrolled a cohort of consecutive patients who are critically ill on the breathing machine, and there are 851 of them, and it was a study that was decline funding by what was our Ministry of Health Ontario at that time. We submitted a grant for about $480,000, and they sent it back and said, okay, you received a grant, it's $48,000. And so I remember calling them, and it was in the day when people actually answered the telephone, and I said, is there a zero missing there? And they said, no, we think it's worth about $48,000, not $485,000. So we didn't know what to do, but we decided to go ahead and try to use the money, and people did this study for free because they knew it was sort of important. And we just observed the ventilator is weaned off, as you can see, in 538 of those patients. So the person got better, breathing tube comes out. Still, there's some patients died in the ICU, some people died on the ward after, and some people went home, 90% of them. So in other words, this is sort of the good arm, if you will. And then there's a bunch of people who died on the breathing machine. And then there are people who had ventilator withdrawal in anticipation of death. And this is how most people pass away today in an ICU. It's following the withholding or withdrawing of some kind of advanced life support. And this was of great importance because the way in which this was done and from whom life support was withdrawn was not studied properly. No one wanted to talk about it. In fact, people wondered if this was, not only appropriate scientifically, as was suggested to me, but wondered whether this was really ethical to study this. And I thought, ethical? This is unethical not to study this. And we simply observed that when 166 people who had the healthcare team, obviously largely driven by the doctors, were saying, we suggest withdrawal of life support, so-and-so is not going to make it. And you can see after the withdrawal of the life support, and this was all ventilation, the 87% died, but some went to the hospital, and some even left the hospital. So suggesting we don't always get it right. So generating data that is either unwelcome, uncomfortable, or counterintuitive is also an important approach. And we went on to say, well, who is it who has ventilator withdrawal, and what are the determinants of that? Because you might imagine there would be like the sickest people. That would make sense, right? The sickest people, the families who discuss, oh, so-and-so is so unwell and probably won't make it. But it doesn't work like that because these are the data reflecting the most important independent determinants of withdrawal of life support for those on the ventilator. And one is the life support of inotropes and vasopressors, which is a kind of life support that makes your heart keep going and keeps your blood pressure up. So if you're on 2 life supports, that would be a marker of someone who might be approached, the family might be approached for withdrawal of life support. That might make some sense. If the healthcare team predicts less than 10% chance of this person going home, okay, that makes some sense. If the healthcare team predicts very poor cognitive function of this patient, maybe that wouldn't be wanted by the patient or the family. Or if the family tells us that this patient really, at this time, with all that I understand, would not really want to keep going on life support. So instead of all the biological determinants of old person, super high illness severity person, 100 comorbidities, those were not the determinants. There were more probabilities, perceptions, discussion-oriented criteria, along with the inotropes, the second kind of advanced life support, that was the determinant. And this turned out to be a super important publication in the New England Journal of Medicine, paid with $47,000 for this international study just to observe this phenomena. And we went on to ask people what their thoughts were, the healthcare providers, and when there's someone who is doing very poorly, and you can see a range of concerns of the healthcare providers. It looks like people are overestimating the probability of survival. Is it really appropriate to use healthcare resources for this situation, which was quite controversial to find. But clearly, wanting to make sure that the care we give is concordant with what the patient would say if she or he could speak for themselves. So it just shone a light on this. And this research program was sort of on a slow burn, I'd say, and for 20 odd years. And now end-of-life care is a much bigger deal. But back then, it was a super helpful program because it engaged everybody. All the healthcare team had feelings and thoughts and observations about this, especially the bedside nurses who are providing the minute-to-minute, hour after hour, one-on-one care with the fragile, critically ill patient. And it helped us be self-reflective, which is a big part of practice these days, and helped to legitimize end-of-life research, I would say, and turned into a number of, shall we say, traditional products. So for this research program, this passion that I described from my own practice and my own teaching, the world wasn't really ready for this to be a research program initially, and I just had to be patient and eventually persevere. And in the time of COVID, our program, which is, as Brittany mentioned, our Three Wishes Project, which is designed to try to honor the inherent dignity of people who are in the ICU when they can't speak for themselves and they're so very vulnerable. It's also to help families to ease their grief a little bit. It never deletes it, obviously, but just to try to humanize the dying process. You saw the image, and some of you have been in an intensive care unit. It's pretty stressful. And just to try to call forth clinician compassion, which is one of the major reasons, obviously, people go into healthcare. So through some of these elicitations and implementations, we're really always hoping to try to bring some comfort and some meaning. And the idea is we got more and more and more technology in my field. Humanism has been, from the perspective of many, a casualty of the pandemic. And so what I'm always trying to do is ensure we retain, if not enhance, dignity, conserving care. And these are just some of the categories from a slide that my good colleague France Clark has developed to just classify the different kinds of wishes of family care, offering tea and biscuits for the family, sitting there forgetting to eat, bringing a pet in from home, paying it forward, even organ donation for those people for whom that's something important. That's a possibility. Readings, rituals, religious rituals, but most of these are secular wishes. Music therapy, all kinds of connections. And these activities have really been very, very helpful. And we've got 26 different publications associated with this, and our colleagues from coast to coast, actually. And our interview data of family members, because we can't interview the patients, they've died. Our interviews of clinicians would clearly show their expression is they are, in fact, calling forth their own sense of compassion and enhancing their sense of responsibility and remembering why they went into healthcare, but we're also trying to help give the family a voice. And this was super, super helpful in the pandemic, where we learned to remember who it is in the bed before and beyond their critical illness, and just try to fill in the gap when the family members couldn't be there at the bedside. Interestingly, the group of high school students this morning were commenting on the use of technology and the FaceTime and different approaches with video conferencing that ended up being an important mode of connection in the pandemic, and it's one of the hangovers, legacy, I would say, of the pandemic that I hope will continue to use, but not in an elitist way, because there's very many disadvantaged communities linguistically, culturally, people without the technologic expertise, visual, auditory impairments. There's a lot of people dialed out of this opportunity. So we have to be careful about our technologic connections. I'll just leave you with this slide. Maybe something here resonates. So I think in the future, we're going to have more and more technology. There's no end in sight in the ICU. But I'm hoping we'll do this. So it's been a great Gairdner Week, and we don't do enough celebrating in science. But I'm not here celebrating anything I've done, but celebrating what you all are doing and honoring the process. And hope you can knock it out of the park on that little icon and keep going, iterate, improve, and pay it forward and celebrate. Thanks.
So we do have time for a few more questions. They'll bring
Thank you for a really beautiful and inspiring talk. And I was thinking a little bit about the relationship of the first half to the second half. And I was thinking a little bit about the definition of patient, and especially at end-of-life care. How much should the family be thought of as a patient? Have people taken this to kind of questions like does end-of-life care, especially for a coma patient or someone else that's not going to regain consciousness before end of life, how might the actions taken toward that person in a healthcare setting affect, say, rates of depression among family members or mental health of family members? This seems like a place where we really deserve RCTs so that we do right by kind of the extended patient in this setting.
Yeah, that's a great question, John. The follow-up studies that have been done of surviving patients and their family members show higher than, shall we say, population norms, maybe not higher than expected, but high rates of depression, PTSD, even caregiver burnout, very concerned about loss of mastery for individuals who maybe are not playing the role in the family they will have played, gone back to work, financial concerns. There's a lot of reality-based consequences to surviving critical illness for that patient who was always a person and is a person, and the associated family members. And so we have now an emergence of clinics, sort of post-ICU clinics that are offered which Margaret Harridge, actually here from Toronto at UHN, has spearheaded beautifully to try to understand more holistically the impact of coming through critical illness for the patients and their families as a dyad, family, not just one, but however family is defined, and the patient or the person. So there's a lot of work identifying the problem, just as we start with observation, what is going on, and now more and more studies we can't necessarily prevent it, but we can hopefully mitigate it, minimize it, and offer supports as needed from whatever dimension of health in all of its breadth need help. And this has only been maybe in the last 10 years it's really, really come into play, but the last 20 years. For patients who die, the family members left behind in grief have, as you might imagine, also high rates of PTSD. People can't come back to the ICU. They can't even drive by the hospital. They obviously are in grief. And like anybody who's grieving may have complex and protracted grief. And the bereavement services are very poor afterwards, very weak. It is not necessarily the stiff upper lip, white Eurocentric. It shouldn't be that way in Canada. We're more multicultural, more diverse, but there is a real inattention to it, and not turning a blind eye to it, but insufficient resources for bereavement counseling. And we're in our hospital working hard on that, acknowledging the important role of spiritual care, psychological, and often it's limited, even grief counseling. But it even starts before patients go home or before patients die, because that's the stress we're seeing and the angst, and that's part of the moral distress of clinicians, seeing things unravel and bearing witness to what is going on biologically with the patient, but psychologically, socially with the family. So it's finally, it's in the press, let's say, it's in the news, I'm sure, but we're attending to it more clinically, and I hope that research will catch up, because I think it's very much needed. Thank you for your thoughtful question.
So there's a very excellent talk, how you shared not only your successes, but the challenges of doing, clinical medicine, critical care, and doing research in Canada. My question is focused on using evidence-based medicine. You talked about your mentors. From your PROTECT study, you had a high-profile publication showing the benefits of use of low molecular weight heparin in cohort patient. Has that translated into common practice in the ICU? And if so, how long did it take? So our first speaker, Dr. Jumper said, AlphaFold2 came out 2020, and how many millions are using it today? I don't know if that's common in clinical or medicine, but in terms of perspective, when something really can change the outcome of patients, when does it become common practice?
It's also a great question. Practice takes a little longer to be modified for many, many different reasons. I think it's faster today than it was a few decades ago. But sometimes if there are mitigating factors like cost involved, it may take a little bit longer, or in this case, generation of evidence showing the cost effectiveness, which was a big game changer. So after a trial, many centers had shifted over to low molecular weight heparin, but there wasn't and we did a whole post-trial knowledge translation sort of research program I didn't go into, but one of the things was asking people, because it's not my business to try to make people do anything, but I wanted to understand what were the parameters, and a lot of folks were concerned as stewards of the healthcare system resource profile. Should we be using it if it's more costly, if it's not significantly better, but it's a little bit better? So there's some concern about the economic evaluation, which is why we generated the evidence about the economic evaluation. And a lot of things in systems in life, as you all know, there's multiply redundant systems that get messed up when an error occurs. There's also multiply redundant sort of barriers or bumps in the road, which could involve the preferences and habits and pre-printed orders and what pharmacy has on hold and are there any guidelines, and there's so many different factors. So things have sped up in terms of knowledge translation and application. And I think they've accelerated enormously, witness the COVID research, and there's still south of the border, I think the data show more Americans got hydroxychloroquine and ivermectin than got other helpful interventions for COVID. So there's many mitigating factors, but I think in general it's accelerating, but it is, we're looking for enabling and transformative ways to make that happen. But honestly, in the hospital system, a lot of it is driven by pre-printed admission orders and things like that. And I think the accelerated approach to evidence integration now is at a jacked up time, very, very much after the pandemic. In certain sectors, it's already well-oiled machine, maybe cardiology, for example. But yeah, there are a lot of moving parts, but I think I know we're in a much better place now than even 10 years ago or even 5 years ago in terms of evidence application, more evidence-based guidelines, and yeah.
I think we have time for one last question.
Thank you, Dr. Cook, for the sobering presentation. Earlier, you mentioned some particularly Canadian challenges, primarily that of low population density, low funding, and sometimes insular communities. Do you find that this is even more exacerbated in rural communities? And if so, what are the particular steps taken to address these concerns?
Yes, thank you for that. There's an initiative in Canada in critical care led by Ali Binney and Jennifer Sang in Toronto and Niagara, respectively, focused on exactly your issue, which is trying to democratize research options for patients in sort of outside the classical quaternary care, big GTA centers, and make sure that research is able to be accessed in centers that would be, great community hospitals, community academic hospitals, in mid-size and even smaller communities. So we have a big network they've started related to community hospital research, and it's just taken off. So Brantford Hospital, Niagara Hospital are two of the most active community hospitals, enrolling tons and tons of patients, offering research opportunities for the critically ill patients serviced by that hospital. And of course, once they are enrolled in those studies, the data generalize much better to diverse Canadians in diverse settings. And then the fantasy and hope is that the results of those studies would be more, front and center in the minds of the practitioners in those centers. So, and then there's something called the Accelerated Clinical Trial. It's an initiative PJ Deborah from Hamilton and Guy Rilot from Montreal have started, and they've identified some what they call portfolio hospitals in Nunavut and Nanaimo and Cape Breton, Nova Scotia, for the same reason, to enhance the capacity and research infrastructure so that research carries on in those places, and when the studies are done, the results are more readily available and used, shall we say, in those communities. So critical care is taking this issue you've raised very seriously, and you may know that most Canadians are cared for outside quaternary care, urban medical school affiliated hospitals. And our research has to mirror that, where the patients are, for reasons of access, availability, and obviously generalizability of the results to all people across the country. So there's quite a bit going on in acute care, especially critical care, in the last 10 years. So thank you.
Okay, so thank you very much. Before we finish here, before we finish, Jennifer will say a few last words and just a couple of quick things from me. Save the date. A year from now, we're back here again for another exciting program, guaranteed. Also, I know people have to leave very soon after Jennifer says a few final thank yous and words. But I think our speakers have a few minutes, not very long, before airport trips and various things like that. So there might be some time for a little bit of person to person, or just sit around and talk for a few minutes. Not long, unfortunately, but we may have some time for that. So, Jennifer.
Thanks, Ron. On behalf of York University, I would like to thank our distinguished and esteemed Canada Gairdner Award laureates, Dr. Deborah Cook and Dr. John Jumper. Thank you for coming to York today to share with us your outstanding research. It really has been a pleasure to hear about these diverse research areas of research and your contributions that you've made to biomedical research and the global health industry. Also, thank you to our audience and guests today for coming out and taking the time to attend. And I would be remiss to not thank our sponsors for today's event, without which it could not have taken place. Thank you to the Globe and Mail, the Canadian Institute of Health Research, the Government of Canada, York University, and the Gairdner Foundation. And finally, thank you to all of you who have worked behind the scenes to make this event a success. And on behalf of York, I do have small tokens of appreciation to both of you for the time that you've taken to come here. Drs. Cook and Dr. Jumper, apparently underneath here, there are some magic bags with York tokens in them. And as Ron had said, if you can stay behind for a little bit, there can be some more discussion about the science that you've done. It really has been wonderful to hear about your work, but also to hear the engaging questions from the audience. So thanks again, everyone.